Hey friends, welcome to this channel. If you're new here, I am Indrani from Bong Biology, and this is Endoplasmic Reticulum part 3 video, where we are continuing on our discuss about how protein is being sorted via endoplasmic reticulum. So in previous videos we have talked about the signal sequence and the secretory pathway, and in this video we'll discuss about the transmembrane proteins. So in previous video we have also shown that there are three kinds of orientation. One is if when N terminus is present on the cytosol, C terminus is present within the lumen. The second one is where N terminus is in lumen, C terminus is in cytosol, but for both these cases there is only one transmembrane part. And the third type is, this is one type, this is second type, and the third type is when we have multiple transmembrane domain. So how these things happen, how they are incorporated in the how this transmembrane part is incorporated into the membrane of the endoplasmic reticulum. We'll discuss about that. So normally what happened in case of secretory pathway or secretory protein is the N terminal should enter first. I think you remember that this N terminal always contain the signal sequence. So N terminal will enter first. So N terminal will enter first, and then signal peptide part will be cleaved. So N terminal will remain in the lumen, right? So over here the same thing is happening, that first there is a signal sequence. This is the signal sequence, it is entering, and then the signal peptides will cleave this signal sequence. Okay, but before that, for the case of transmembrane protein, what happen is there is a stop transfer signal. The stop transfer signal means there will be an alpha helix and 20 to 25 hydrophobic stretch will be there. So this is called the stop transfer signal. And when there is a stop transfer signal, after the signal peptide is being cleaved, N terminal is present within the lumen. However, due to this stop transfer signal, C terminus is present in the cytosol. Okay, so N terminus is present within the lumen, C terminus is now present in the cytosol due to this stop transfer signal. So this is one mechanism where we have stop transfer signal. So we have stop transfer signal. But there is always the second orientation, that is when N terminus is at the cytosolic part and C terminus is in the luminal part. How this thing happen is via another signal sequence, that is called internal signal sequence. Now if there is an internal signal sequence, it may happen that this internal signal sequence is again alpha helix and there are 20 to 25 hydrophobic stretch of amino acid, and so this will obviously not enter inside the lumen. So it will remain in the transmembrane part, and as a result the N terminus part cannot enter into this lumen. So this N terminus will remain on the outside, and finally when the polypeptide synthesis is done, the C terminal, because in this case we have in this part we have the signal sequence, internal signal sequence, but over here we don't have the hydrophobic stretch. Okay, so this part will enter within the lumen, but this part will not enter within the lumen, or that part will not become cleaved also. So as a result there will be N terminal will be on the cytosolic side and C terminal will be on the luminal side. Remember, when we have normal signal sequence, this normal signal sequence is always present at the N terminal, but here this is called internal signal sequence, because at N terminal there is no signal sequence, but the signal sequence is present a bit internal to the N terminal part. As a result, this forms the transmembrane domain, and N terminus of the complete polypeptide remain on the cytosol and C terminal remain on the endoplasmic reticulum. Another thing which can happen is somehow if N terminal enters into the lumen, then C terminal cannot enter into the lumen because of this internal signal sequence. So due to internal signal sequence, two kinds of thing may happen. One is C terminal enter into lumen, N terminal remain in the cytosol. It may also happen that N terminal enter into the lumen but C terminal remain in the cytosol. Okay, hope that makes sense. And what happen in case of multiple transmembrane domain is there will be both stop transfer plus internal signal sequence. They will remain as alternative form. So like one stop transfer, then one internal signal sequence, then again a stop transfer, then again a internal signal sequence, like this. So let's say first this is an internal signal sequence, so N will remain outside and C can enter, but if there is again a stop transfer sequence, then the whole C will not enter and there will be a loop formation. So now here you can see that there are two transmembrane domain or two transmembrane part, where the first part is due to internal signal sequence, internal signal, and the second membrane or transmembrane part is due to stop transfer signal. Okay, so this is two transmembrane domain. If the same thing continued, that after this internal signal sequence and stop transfer there is again a second signal sequence, then there will be again three transmembrane domain, and so this thing will go on. So what happened in case of multiple transmembrane protein is there is alternating stop transfer and internal signal sequence. What happen if only stop transfer signal is present is N terminal will always remain in the lumen and C terminal will always remain in the cytosol. And what happen in case of internal signal sequence? Two things can happen. N terminal can remain in the cytosol, C terminal can enter into the lumen, or it may also happen that due to internal signal sequence C terminal remain in the cytosol and N terminal enter into the lumen. Hope this makes sense. If there is any query, please ask me, I will describe. Okay, so this is it for this video. In the next video we'll discuss some more. Okay, before wrapping up this video, one just small thing. So we can see that if there is transport of the secretory, what is the overall topology of the secretory pathway? We can see that the lumen of the endoplasmic reticulum, this is the lumen of the endoplasmic reticulum, and this is the Golgi apparatus. Okay, so the lumen of the endoplasmic reticulum and Golgi apparatus are topologically equivalent to the exterior of the cell. Consequently, those proteins of polypeptide chains that are translocated into the endoplasmic reticulum are exposed on the cell surface following the transport to plasma membrane. So finally they are fused with the transport vesicles are fused with the plasma membrane, and then all the contents are being released onto the cell exterior. So this is the overall topology of secretory pathway. So see you all in the next video.